Okay, so um, for today's new stuff, you'll want to go onto Canvas and get the Colab notebook and the PowerPoint. And I didn't totally forget, I just mostly forgot about finishing up the eQTL stuff. So I'll go back to that in a second. But just to give you the overview where we're at in the course, right? We've got today and then next week for machine learning. And then the plan is to do a single cell RNA-seq data day and a metagenomics data day. We're going to see, just because we're at least one class period behind where I thought we would get to. It's fine. It's just, that's part of the reason the assignments have been late being posted, because I don't know what material we're going to get through. So what's going to be the relative assignment, the relevant assignment. The assignment for Monday is going to be another image upload, just like the last few have been. It'll be related to the Colab notebook that we have for today. So what that means is there's no homework related to the eQTL notebook. So let's go finish up that eQTL. Actually, I'll follow the link from Canvas because I don't remember which. Right, so it was this one. I had all kinds of problems with this notebook, so we'll see if it runs. How many of you were able to finish this notebook or tried to finish this notebook? Yeah, did it work? Oh, good. Yay. So not that many, but one thing is that I didn't really anticipate is how slow Colab can be trying to do the regressions. So trying to do, like, even if you put it on the minimum 200 by 200, it takes a while. And then that's why the last few cells actually skip over and just jump to downloading the GFF file for the Saccharomyces genome, and then getting the data 9 from the actual paper. Should have run this before class. So what it's doing, I'm sort of skipping past where we got to on Tuesday, which was doing the regressions, sort of building that variant by expression and identifying the eQTLs on your own. What this is going to do is get the genome for Saccharomyces, just so we have positional information. And then if you go back to the paper, they have the matrix of genotype by expression value, eQTLs. Those correlations are already done in a matrix. So rather than just doing the 200 or whatever that we would have been able to do in class, now we're zooming out and just taking their computation for all of them, so that we're not just limited to looking at the subset. Data 9. Okay, and like I alluded to on Tuesday, we get something that looks like their barcode chart. That's what I called it. This isn't quite like theirs just because of the way that the Colab notebook samples, just to make it a little faster. I think I limited it to the top 3 genes, the top 3 correlations for any variant, something like that. And then the dot size is scaled to the matrix. So I'll open up just, whoops, oh, it's so big you can't. Okay, so just to give you an idea, I keep talking about this, but I know it's hard to visualize. So this is the data 9, and so what it did, right, is these are all the marker positions in columns and all the genes in rows. And so all the values here are the correlation between that marker and the expression of that gene in the, depending on its genotype, right? And so, like I said, that's sort of what we got to on Tuesday. This would have been the output of that analysis. You can think of these correlations as the regression coefficient, which we're going to get to today. But then what this figure does is take all of those associations, and I'm just pulling out like the top 3 values for any marker. So that's why it doesn't plot every single one. And then the size of the dot is how big is that number, right? So if it's got a bigger number, it gets a bigger dot. So how do you interpret, back to the question about biology, what are we trying to do with the eQTLs? We're trying to predict which markers affect gene expression. All right, those are the traits. How does the variant affect the trait? And we talked about cis versus trans. It's a major question when we start to get into functional genomics. How much of the regulation of a gene is due to the promoter region versus transcription factors in other places in the genome? What does this figure tell you about that? I know it was only 2 days and no weekend since we looked at this, but yeah, so what does this figure tell you about cis versus trans regulation? And I mean, even what is the x-axis here? We can start there. So both of the axes show the chromosome number. Right, so yes, I know it says eQTL position. What is that? It's not the most intuitive label. That was partly just to make me think. Yes, it's the SNP position, right? So in that data 9 folder, right, in this folder, it's the columns. So it's telling you the chromosome and the position. So each dot here corresponds to a SNP position, And so then what's on the y-axis? The gene that, in this case, was either up or down regulated. Okay. So to get a dot in this plot, it means that that SNP had a significant association with whether the gene on the y-axis is up or down regulated. Okay, so put those two things together. What is this diagonal line in your board? Now, I drew the diagonal, or had the collab notebook draw the diagonal line, but why? What's significant about cis versus trans? What's the diagonal line? It's the cis regulation. So it's like, if you're on that diagonal, it means you're in the same position in the genome. That means the marker and the gene that's being expressed are in the same position. Those are the cis regulatory SNPs. So what are these weird dark bands that are vertical in this graph telling me? Trans regulation, yeah. So there's some SNPs on chromosome 14 that basically affect the expression of all the genes in the genome. I mean, that's an exaggeration, but because the dots are big enough and there's so many of them, it looks like a solid line. So we would call that a master regulator. Right? There's something about the gene where they're like, this is a transcription factor that mutation, whatever the marker is at that position, is probably changing the conformation of that protein. And that protein is a transcription factor that regulates a whole bunch of stuff in the genome. Okay? So hopefully, think about it, look at that paper. This is something in terms of the biological interpretation of what you're doing that's important to internalize. Because this is a very cool way to get at the functional genomics of what your SNPs are doing to the transcriptome, or how they're affecting the transcriptome. Okay. Any questions about that? Like what eQTLs mean? Okay. So with that, any questions about that stuff? We're going to move to Intro to Machine Learning. So Thursday, there are 2 links. There's a PowerPoint, not too long, and then the collab notebook, which is what we're going to spend most of our time doing. So some of the stuff we talked about a little bit before, way back at the beginning when I was talking about generalized pre-trained transformers, like what is ChatGPT. I gave some background on that. So AI being, actually, I'm getting ahead of myself. I've got that Venn diagram sort of in a couple slides. These are the kinds of things that I'm hoping to get to. Today we're just going to very high level overview what is supervised versus unsupervised learning. And then my goal for today in the collab notebook is that if you don't understand regression and principal component analysis, sort of how those are related, I'm trying to build some intuition for that, because lots of what we do downstream in machine learning is based on that foundation. Now, you can take a whole class on machine learning. In my PhD, I had a whole semester just on regression. And so we are not going to get into the weeds on the math, okay? So, but my goal there is let's just build some intuition about what the models are doing, okay? So what is it? We talked about this before. Machine learning is the data plus the algorithms. And in some sense, it's teaching itself. So enabling the computer to sort of learn from patterns that are in the data. So it's those algorithms that are the learning part. The data is sort of what you're training those algorithms on. So the three things are that the algorithm is learning from the data. Ultimately, you want it to be generalizable, okay? And then typically there's some kind of feedback loop that's necessary for the learning. The reason that I'm going to talk, and then we're going to spend most of a whole day talking about regression, is I think it's the easiest example for how all of these things are related. Okay, so this is the Venn diagram I was talking about. Artificial intelligence is sort of the umbrella for all of it. Machine learning is this subset where we're trying to come up with algorithms that help learn from patterns in the data rather than hard coding the rules. And so I gave this example at the beginning of the semester, like what are those things? Most people would call them a mug, okay? But then, the Starbucks cup here, is that a mug? I don't know, but how do you know? Like, what's the rule, right? Tell me what rules define some of these as mugs and some as cups, okay? Similar thing, maybe a more fun example is you can hard code. It's not that difficult to write a program that knows how each of the chess pieces can move, right? That doesn't mean the computer can play chess, right? Even if you get sophisticated about interactions between pieces, it still doesn't really teach the computer how to play chess. At least it's not going to be able to compete with a person. And so then how do you get to, this is the graphic for, I'm not enough into chess to know how the rankings are computed, but the solid line there is sort of the distribution of people who are into chess enough to have a ranking. It's like if you're into golf enough to have a handicap. Then, like I guess in that group of chess players, the average is about 1,500. The best ever human ranking, it's too small for you to probably read. Anybody know who it is? Who's the best chess player in the world? Gold star for you. Yeah, and his ranking, his best ever ranking, which is from like May last year, was about 2,800. The best AI chess engines are in the 3,700 range. Like there is no person that can compete with a modern chess engine. Like it will win 99 out of 100 games. And so that is way beyond just coding how do the chess pieces move. That is being able to learn what good board positions are based on basically analyzing every chess game that's ever been played and then playing against itself and seeing what positions win. There are tons of information about how these things work. It's a rabbit hole that I went down when I was making that slide. Really cool stuff for machine learning, though. there are, I mean, some depends. Some people say there are two general approaches to machine learning. Some people break it down into the four. People who break it down into two, they're just supervised and unsupervised. So supervised is when you have some labeled examples. When we talked about GPTs, right? When you thumb up and thumb down ChatGPT, it's learning what is a better answer. That now becomes labeled data. So ChatGPT is, though, probably best put down in these sort of semi-supervised because it can understand lots of things from context that are just based on a previous model that had supervision. Unsupervised, when there's no labeling in the data at all. This is, we'll see the distinction here when we talk about regression and PCA. But I've given you a few examples there. The things we did with predicting, or we will do, predicting tissue types from gene expression profiles. That would be a supervised thing where, think about it as a marker gene. If you know there's a marker for a certain tissue type, you can give it all the expression data and say, when you're trying to define the tissue type, give more weight to this marker gene because we know that that's associated with a particular tissue. Okay? Whereas unsupervised is just saying, in the case of microbial communities, I don't want to make any assumptions about environment or host or any of that stuff. Just tell me what are the clusters of co-occurring microbes. So semi-supervised, right? That's sort of my labeled cell types example that I just gave. Whether it's supervised, this is why are there two or are there four? I don't know. Not super important, but I put it in here because it's common in the machine learning literature. So, and then reinforcement learning is that's maybe a better example of what ChatGPT is doing by the thumb up and thumb down. We're not going to get to all of these, but I wanted to give you the vocabulary. All right, so if we were doing a semester of just machine learning, this is the list that we would go through. We're just going to do logistic regression. Next week, we may do random forest and support vector machines, but we'll see how it goes. Still trying to see if Collab can keep up or if we need to move to bridges for real human data. Okay, so we're just going to cover the first one, applications of this. Okay, so this is not the chess picture again. This is, there's a lot of research going on, a lot of people working on how do you take various kinds of imaging data and make diagnoses. So this is the example for if you ever have pneumonia and they take a chest x-ray, you have to wait several days usually at least to get your results back. And why is that? Because there's a person that has to look at that x-ray and try to make a diagnosis about whether you have pneumonia or not, right? Maybe it's just bronchitis. So it would be a lot more efficient if a computer could look at the x-rays and make the diagnosis. And so the green line here is, the scoring is sort of arbitrary, but if you take the average radiology score in terms of verifiable, this is the image, they did have pneumonia, versus the best machine learning algorithm for this right now, right? It's doing better than maybe 70, 75% of radiologists. Okay? So that's not perfect, but sort of like, it's not at the chess engine yet where it does better than every person, but it's doing better than most radiologists. And this same kind of um, approach is being used a lot in cancer, and basically anything you can think of that takes an X-ray or an MRI or an ultrasound, trying to train a machine to make those diagnoses will speed up the path to treatment, hopefully. Like, that's ideally. Okay, so that's hopefully a little bit of motivation for why we're going to go try to build some intuition about what's going on in machine learning. All right, so first cell, just import our tools, NumPy. We've used several of these tools before. In fact, we've used these approaches before. We've done regression several times already in genome variant analysis and QTL analysis or eQTL analysis. We just haven't talked about what's going on when we import these things and use their functions. So this is just basically NumPy is we're going to use it for random number generation, some tabular stuff, but that's mostly pandas, which gets imported in the second cell. And then the plotting stuff is in matplotlib. Here, we're defining this variable RNG that you'll see several times when we simulate data, and this is just trying to make the data that you guys generate with your collab, basically everybody's looking at more or less the same standard data. And something about when it shows, we're going to only show 4 decimal places, for example. Not super important stuff. All right, so now the second step, we're going to create a synthetic data set, and you should see this linreg underscore data. So it generates a CSV file. It should look something like this. Right. So we've got an X variable and a Y variable. It's sampling that from a normal distribution, sampling uniformly from -3 to +3. And then it's generating the Y data based on this equation. Okay? So I skipped over the error part, which is important. So it's adding some error into the data so that it's not perfectly on the regression line. So just to make it look a little bit more like real data. If you wanted to add more, like later at the end of the day, I'm going to say, go play with this a little bit. And you'll have to play with it for the kind of image I'm going to ask for in the assignment. Not necessarily this cell, but understanding how these cells work will be needed. So you could add more variation if you want. And so here, if I didn't already have this one commented, the comments get less and less as you go down on purpose, right? So here the training wheels are on, later they're off, okay? So, right, how does error get generated here? It's using that RNG, the random number creator, sample from a normal distribution. This is a function that will pull from a mean of 0 with a standard deviation of 1, and then it's using n, which we defined as 100. Okay. If I didn't have that on there, you could ask Gemini, what does this line do? Or you could copy and paste it into ChatGPT t and say, what does this line do? And it should tell you what that function is doing. Hopefully that's what you've been doing a lot lately. Okay, and then the second part there is what writes the linreg underscore data so that we can use that later. Okay, so this graphic, unfortunately, doesn't use the same notation, but this was the, I don't know, I need to find a different picture, but theta 1 here and theta 2, later I'm going to call them beta 1 and beta 2, or B1 and B, or B0 and B1. Hopefully you can make the connection. Question is, why is linear regression considered a supervised learning approach? So if you've taken basic stats, you probably didn't call linear regression machine learning, okay? And this is why sometimes old codgers like me say all this machine learning stuff is just multivariate statistics repackaged, okay, with big computers, right? Just given cool buzzwords to old stats. Not 100% true, but you know, on my cranky days. Why is regression a supervised learning approach?
Because you're giving the target like the y along with the training data.
Yes, right? So the x is like the training data, and y is the sort of known target. So you've labeled that value of x with a y value. And so you've taught the computer that given this value for x, this is what you should expect for y. That is machine learning in some sense, right? At the most simple level, the computer can now predict values for y based on x. So if you give it new data, it will spit out new x data. It will give you new y values for that, okay? So because I knew not everybody had stats, I tried to go halfway between getting into the weeds of the math and just showing the examples for the intuition. Lots of times in the machine learning literature, you will only see matrix or the linear algebra expression for how you compute regression. So I just have a couple cells here showing the connection, because in biostats and biometry probably, you see the scalar notation for this, right? So this is the most common one you see is y equals mx plus b, or so this is saying the predicted y is equal to the intercept, beta 0, plus beta 1 is the slope, times your x value. To solve for the slope, right, we're just taking each of the x values and subtracting it from its mean, the mean of all the x values, multiplying that by the difference in the corresponding y value from the mean of the y values. Okay? So that's taking each of the x's and subtracting it from the average of all those x's, and then doing the same for the y's, and multiplying those together and summing them. Okay? And then the bottom, anybody know what that bottom is called? Anybody recognize it? It's like one of the most famous equations. It is the variance, yes. And variance also often gets called the mean square error. Okay? So it is taking the difference between any individual x minus the mean of the x and squaring it. All that squaring is for is get rid of the directionality, okay? And it's summing all of those. Anybody recognize the top one? Got some data science students in here. What is the top? This is the gold star question. Actually, variance was pretty good. It is the covariance. Very good. So slope. So it's another aside. Some people think better. Learned this during my PhD too, because my PhD advisor would get up at the whiteboard and start doing equations all day long, and everybody in the lab would try to hide behind the monitor so we couldn't see how puzzled we looked. And he would stop every once in a while and go, clearly this shows. And like he just thought in math. I think in figures. And so, but all that is to say, people have different intuitions based on the equation versus the plot. Okay, so the covariance divided by the variance is the slope of a univariate regression. And then once you have that, you can compute the intercept there just by filling in and rearranging. Okay. Now, all of that is the same as what I was saying before. In the machine learning literature, you see this reduced to the matrix notation for it, partly because we're almost never dealing with one x variable when we're doing machine learning. We've got tens, hundreds, thousands of x variables. And so this is what that notation looks like. So you've got some vector of x or matrix of x. In this case, right, it's a 2 by n matrix where you've got the first column, that's just going to represent the intercept. And then the second one is what you're going to use to calculate the slope. You've got the vector of betas. These are the slope and the intercept in the case of a simple univariate regression. You'll see later when we start to talk about PCA, these are analogous to the weights. Like how much weight are you going to give to something? That is the slope. And then y, the vector of your results or the thing that you're trying to predict. This equation, that when you see the term ordinary least squares, usually they're talking about this equation right here, y equals x beta. Sometimes they include the error term in it or not. Okay. And so what you're trying to do is minimize the difference between the y vector and the multiplied weights times the x is sometimes called design matrix, but that's the data. Okay. That minimization, if you write it out, looks like this. Again, linear algebra is not a prereq for this class, so I'm not going to go into the linear algebra, the matrix math. But if you solve that, that is what people call the closed form ordinary least squares estimator. Okay, so if you're going to get the exact value for the slope and the intercept in univariate regression, that's what the computer is probably going to do. Okay? The reason some of this like closed form, have you ever even heard that term before? Yeah, probably not, because it doesn't get used outside of machine learning that much, or I suppose if you're in the math stats, they may talk about it as well. But all that means is you're getting the exact solution. You're not doing any kind of iteration or search. Okay? This solves for the best value of the slope. This is just showing you that they're actually the same thing, right? So those terms, the transpose of x times x, turns into that little matrix. Transpose of x times y turns into that vector. And then, right, you're multiplying those two, this matrix times this vector gives you the expected weights or the expected betas. Those are, right, the same as the equations up here for beta 0 and beta 1. Right. Sorry, I should put all these in one cell so you don't have to look all over where they came from. I've already talked about these. So that is the covariance divided by the variance. It doesn't matter how many x variables you have in that design matrix. When you get multiple variables, it's no longer a single line. You have two variables, it turns into a plane. Okay? When you have three or more, they call it a hyperplane. Okay? That's just the jargon. Because this idea of minimizing the slope by taking the covariance of whatever x variable you're talking about and dividing it by the variance of that, you can do that to any number of variables. All right, so let's draw a picture of it. What you'll want to do here, so I've got markers saying what the different sections are doing. And you can, I left the code as close to the equations up here as I could. Right, so calculating V0 is the y dot mean, computing the mean. Right, so it's doing the intercept. Okay, so if you go down, what you'll see at the end here is that first it plots the data. So this is our data that came out of that linreg underscore data. It's simulated. Ordinary least squares solves for the slope. And so what this plot did, we scroll up here, we've simulated a bunch of slopes that don't fit the data very well. And so down here, the beta 1s, we just simulated a whole bunch of them. And I don't know, simulated is the wrong word here. We've plotted that function. Okay? And it's just showing that the slope, 3.51, is at the low point of the mean squared error. So and then the last one shows you drawing that slope onto your data. And so if the math is not very intuitive, what's going on here is that that minimization function, that the x transpose x times x transpose y, it is minimizing the distance between each one of these dots and that line. So that is, if you were taking that mean, that is the mean squared error of this regression.
Is there a particular reason you're using MSE here as a metric? Because I think later on you use far MSE. So then that's like interpretability to the rest of it.
No, there's not. Did I switch to RMSE later? I'll tell you when I get there. It'll probably jog my memory when I get there. It's a good question though. Okay, so that's the ordinary least squares solved by the sort of normal probability statistics. That's the way you learned it. The problems, I'll get to that because I've got it in another cell coming up. The way that you will see it solved, and you don't do it for one x value very often, but one of the machine learning approaches to finding the slope is you could sample, you could just keep trying different beta 1s over and over again until you find out which one gives you the smallest mean square error. And so that's what's being expressed there. If you run the gradient descent here, and just lots of sides today. People often think of gradient descent as sort of the machine learning that's going on here. That somehow having the machine test the different betas until it gets the best one is sort of the machine learning. And I don't want to get bogged down in the semantics, but really the whole regression process, finding the slope is at the end of the day what you want to have learned. Somebody tell me what's going on in that figure. How about that?
So the step sizes are like how many like tries basically that the model has gone through.
Yep.
And then it's learning more each time, so then it's getting closer to like the true closed form values. So then you're at least hopefully you're getting better and better results as the steps get longer.
Yep. Exactly. So it's just like I said, it's plugged in the betas, reran the regression, and then this is just plotting them every time. So it started out on the blue one, 0. Got better with the orange one. That's step 100, step 700, 1400. So it's getting closer and closer. This is a plot of how much better, how fast did it get. So the mean squared error is on the left side. So step 0, its first try, it just plotted a horizontal line. Doesn't fit very well, so the error is huge there. So then it kept trying and trying and continuing to get lower and lower because it's getting closer and closer to that closed form solution, right? Of course, it doesn't know that it's getting closer. It just knows that it has less error after it's done. Okay. Some of the reasons that actually, those two lines at the bottom, did gradient descent do a good job or not? Well, do a good job is subjective, right? What's the problem here? Anybody see any problems? First of all, you know the true slope in this case, right? Because you simulated the data. So what is the real slope? That's the intercept. Beta 0 is the intercept, beta 1 is the slope. This is the ordinary least squares estimation for beta 1. The gradient descent is 3.067. What is the actual true value? So we go back up to the top. This is what we simulated, 3.5. Okay, so ordinary least squares is very close. Why isn't it perfect? Why isn't it exactly 3.5? Yeah, because we added error. Okay. And so now back to my original question: did gradient descent do a good job? I can't tell if the silence just means you're unsure or it did a meh job. Meh. All right, so how do you think, let's see where our intuition's gotten so far, how could we make gradient descent do a better job?
Changing more steps.
We could try. So let's find, let's give it 7,000 steps. It's pretty fast. So now, right, by 2,000 it was only there. And look, it got a way better estimate with 7,000 steps. So that's one way. The other way is, I'm going to change this back to 2,000. ETA, I don't actually remember what ETA stands for. I think of it estimated time of arrival. And that's a, if they named it that, it's kind of cool because of estimated time of arrival. You can also think of this variable as how fast are you going to let it learn? So really, it's a scale. It's a how big of a step am I going to take? So instead of taking more steps, let it take bigger steps. Okay. So let's take a 0 out of the ETA and rerun it. So now I've gone back to limiting it to 2,000 steps, but whoa, all of our, like it still started at 0 and now they're all on top of. So it, here we've got to zoom in, but it only took it, maybe 50 steps. It got down to flat at 500 steps because we let it learn faster. Okay. Okay. And it arrived on those big steps even closer to the closed form solution than the slower learning with 3 times the number of steps. Okay. So, 2 ways that you can modify gradient descent to help it optimize the solution. Why would you use gradient descent over ordinary least squared? It's wasting computational power, right? Because you're making it do all of those estimations for all of those betas. Even though it was the blink of an eye, it computed in the 7,000 steps on 7,000 regressions, calculated the mean square error, made the move, recalculated, whereas ordinary least squares just solves that matrix function. Okay. So the problem arises when you get to big data, genomics size data, the eQTL kinds of analyses, because to do the closed form solution, right, that X matrix that you have to transpose and multiply, you have to have all the data in that matrix. And so if your X matrix has lots and lots of variables, or like you've measured every SNP, or it's got lots and lots of rows, that ends up being a lot of memory, and it's not a trivial matrix multiplication step anymore. So as you get bigger and bigger data, sometimes it's easier to just try a vector of betas, see what the average error is, and try it again, because it computes faster than trying to invert or transpose and multiply the matrices. So that's big data and high dimensional data. High dimensional, we just mean more X variables. We're going to talk about ridge regression in a second, but if you need to regularize the data, what we mean by that, like if you have data that are on fundamentally different scales, right? I've got gene expression data and I've got enzyme activity data. Those are not on the same scale at all. But in order to compute a regression using both of them as X variables, we need to put them in some kind of similar framework. That's regularization. Yeah.
A question. For the data grid, do we need to change those numbers for the assignment or for the next? That is something I don't think I remember last year.
It will, well, so if you don't remember what you changed it to, that's okay. You can always re-download the notebook, but I don't know yet. Like, I haven't decided which thing I'm going to ask you to change. Because, well, we have 20 minutes left and we haven't started PCA yet. So how do you know how good? Right, it's going to give you, whether you use closed form or gradient descent, it's going to give you a result, right? It's going to, in the case of univariate, it's going to give you the line that predicts y from x. How do you know if that's any good? So the two metrics that you should look at, the mean squared error, and really the first one I always go to is the R squared. That is the sort of the amount of variance that is explained by your model. So in this case, pretty good. Saying 97% of our variance is explained by the model. You can go back and mess this up again. Force gradient descent to do a worse job. Rerun that cell. So we're back to not a very good estimate of the slope. Rerun this, and you can see that our R squared has dropped, right? That's the indicator. In case this isn't clear, you don't know what the real answer is, okay? And so if I only ran gradient descent, because closed form was too computationally costly, how do I know if it did a good job? Well, what's the R squared value? How much of the variance is your model explaining? Put lots of asterisks next to that, because that's what you want to know. Is your model any good? Okay, all of those cells above, that's my version of computing the regressions by hand. Nobody does that. Okay. At least in the Python world, everybody uses scikit-learn. I should say everybody. Everybody I know of. This link will take you. Oh, that it warns you. Oh, if you click the button, it'll take you here. Lots of models, all that stuff that I had in the PowerPoint, like all the supervised and unsupervised methods. There are methods built into scikit-learn to do all that stuff. Okay, so rather than having to code closed form or gradient descent, we can just say scikit-learn linear regression. Okay. So this is the thing that I love about Python. You just read it. So in this case, we're going to import in just the first line both the linear regression and ridge regression, just because ridge is an increasingly common thing that I see in the literature, and I'll talk about why in a second too. Run this cell. And right, it gave me the same as the closed form. I meant to go look at this. I assume it uses closed form. This is the upside and the downside of scikit-learn. You have to go actually look at the tool if you really need to know. It's such a widely used package for something as common as linear regression. I just trust it. Okay, but I don't know if it's doing closed form or gradient descent with a really steep learn, or if it checks the data first before it decides. I switched to RMSE. It's the residual mean square for PCA because it's what's left after you account for the mean square in whatever the other MSE is. Right? So the other PCA. So if you have principal component 1, it has a mean square error, and so what's the residual mean square error left after you model the second principal component, or the second highest principal component? I think that's what I meant to. I don't know why that just popped in my head, but we'll get there in a second. Okay, so it did a good job, basically giving you the closed form. It's got the same R squared. Point here is don't hand code linear regression, just use sklearn. Or that's just the shorthand for scikit-learn. Okay, ridge regression. It's what I was talking about a minute ago with regularized variables, so that we've got data that's on very different scales. You can incorporate that in ridge regression. Fundamentally trying to do the same thing where you're minimizing this term, which is almost the same. The places where I've used ridge regression is because I know that there are some variables that will dominate the inference. So, for example, got a whole bunch of SNPs, and I know I've got one that's sort of a master regulator, and I don't want that to dominate the weights because I want to see what other SNPs are influencing the trait. So ridge regression will sort of pull the high and the low weights toward the middle. It puts boundaries on how much one factor can dominate the regression. That's sort of my intuitive explanation for what ridge regression is and why you might want to use it. So that alpha parameter is how much are you going to constrain, or how much are you going to force the outliers in terms of weights to be less dominant in the analysis. So it's shrinking the betas, especially the betas that are really high or really low. Hopefully it's the ones that are really low. So this just shows what happens at different alpha. So if you're constraining by a certain amount, right? If alpha is 0, it's hard to see the 0.1 and 1 because they basically all fall on top of each other. You can sort of see difference in 0 and 10 there, the purple and green. But 0.1 and 1 are underneath them. Well, I had it in the intuitions there, but since I've already scrolled past it, why might you want to use this? I gave the example of SNPs, which is where I have used it in the past. Why might you not want one SNP to dominate the analysis, for example? This is a pretty big intuitive leap, I think. But
because if your model is mostly learning from just like one data point or like class, then it's not very generalizable and you can't accurately apply it to like other unseen data.
100%. Generalizability. Yep. So especially in the case of SNPs, you go sample in another population that doesn't have that SNP, and now your model is trash. Okay? So by not letting your model be dominated by that SNP, you make it more useful for other unsampled data. Okay, residuals. In our simple univariate analysis, we did our regression. This is what the residual error looks like. So now it's plotting the distance of every point on the x-axis from its true value on the y-axis. How many of you looked at residual plots before? This is not new stuff. Yeah? Okay. This one is useful, though, when we move to PCA, have something like this in mind. So the residual, this is probably what I knew this was coming. Like how much variance is left is your next principal component. So this would be, for example, my first principal component, and that's how much variation is left over after I've taken the variance or explained the variance from my first principal component. My regression is explaining a certain amount of variation in y based on the x value, but then, in this case, we introduced error, but in real life, there might be some other variable that can explain some of that variance, okay? Or multiple variables that explain some of that variance. Ideally, if you don't see normal or something like normal distribution of residuals, that means there's probably something else going on in your regression, and it's probably not linear. Okay, so that's a key point. So far, we've only modeled a linear relationship between x and y. So a couple summary things there. Optional next steps for you to play with on your own. Go back and play with the simulated data and see how the models respond. You can ask ChatGPT how to do that, how to introduce outliers and see what happens when you've got outliers based on the closed form gradient and ridge. The next thing here is to move to multivariate linear regression. So what happens when we've got multiple variables, multiple predictors. So now our plotted down here is you've got x variable 1, x variable 2, and the y-axis. If I do that, you've got a simple linear regression, right? But that does not really explain all of the variation. So if I was to plot a linear regression here, I should have included these. And then I plot the residuals, what you're going to see is there's a bunch of residual variation left. And I could explain that with the other x variable. So actually they're spread, in this case, in 3 dimensions. Okay. And so in some sense, so it's switched to W here. I thought I had gone back. And when you start to move to multiple regression and principal component analysis, people typically talk about the slopes, the betas, in terms of weights. And so pulling code from different notebooks, and I use weight instead of beta. So just think W's and B's are the same thing. In this case, not good. I use B for the intercept, and then the slopes of the regression are W's. And this model, again, you still use the same metrics. In this case, the R squared is 0.914. It's doing a pretty good job. So this cell will write some new data. Actually, maybe I had that in the previous cell. That's okay. This is another cell just like I had at the beginning of regression to talk about what's going on. This is even more at an intuitive level with less math. But principal component analysis is instead of saying how is X related to Y, we're just going to say what is the direction of the X data that has the most variance. So think about, you spread all your X data out in, like if I've got 5, probably easiest just to think of it in terms of 3, okay? So in 3-dimensional space, if I spread out all my X data points, which direction has the sort of most stretch, the biggest distance between the highest and lowest value in that 3-dimensional space? And then you're going to say, okay, if I put a line through that biggest variance trajectory, that's going to be my first principal component. That's sort of at a very coarse level what PCA is going to do. And so in that sense, it's unsupervised because there's no target here. You don't know what is, we're not even looking for a correct value. We're just asking, is there some underlying, sometimes called forcing, right? That is stretching my data in certain directions. And so here, a regression on just 2 variables would get, actually this is 1. This is the first principal component, basically giving us something similar to a univariate regression. And just to talk about, what's the difference? How are they sort of the same or similar? In our univariate regression example, the error in univariate regression is relative to the y-axis always. So I think of it as you have vertical error always in univariate regression. PCA doesn't know what y is, doesn't care. Okay, so it's calculating error orthogonally. So you can see there are little dots drawn in there where the blue ones are the linear regression error and the sort of red dots are the orthogonal. So that's why error is different between a PCA1 and the regression analysis error. So the regression is giving you a slightly higher mean square error. What I get from honestly, from pulling cells from different notebooks. That's what really that gives us something. Well, last set here. I know I'm skipping over stuff. Just the question there was about the residual mean square again in my head.
Mean squared error?
Is it?
Because it's the square root of the MSE. Because mean squared error, like error is the residual.
Yeah. Okay, I'm going to come back to that actually. So it's the correlation coefficient, not the amount of variance explained, right? The R instead of the R squared. Doesn't matter. I want to talk about the simulated data just so that you know what you're looking at here. I'm going to run the rest of these cells. This is the one. It's not fair. We'll pick it up there on Tuesday. I was going to say that's so where the intent for today was to get to the two-dimensional PCA plot, but I'm not going to skip there and then say, hey, do an assignment on what I had to skip to the end on. So it'll probably be something with the multivariate regression. The details will be in the assignment. Did you already have machine learning as a class? Yeah, I know. Finally getting into stuff that's more relevant.
But is there any, like, because I personally never use MSC because our MSC just seems so much more interpretable and I got a red eye there.
Why you use the square? Because it's still on the variance. Yeah, I mean that's the only reason. It's a pretty variance scale. It's a good question. I don't know why you would. I think, but that could just be again not paying attention. I mean, you do pay attention to. I look for R squared because that is literally the variance explained, and not. If somebody, if you're square rooting the r squared, you lose the directionality.
I'm not talking about squared. I'm talking about RMSE.
Which is the r, but if you just square root the MSE, you lost the directionality of r. So I don't know why. But this is back to, I don't know why RMSE is in that number.
I don't understand. Because how I understand RMSE is that it's just like the average among the residuals.
That you can't just do that because of the directionality problem. You have negative values, and when you're summing, you won't get the mean. Okay. Remind me your name. I know I had you in like human genetics or 1442. Yeah.
My name is Zainab. Yeah. The other one's Malay.
Yeah.